Hello everyone, Mr. Linder here. Let's talk about the cephalic phase, the gastric phase, and the intestinal phase as it relates to digestive regulation and control. So, when you think about the digestive system, you have to understand that the GI tract is integrated in its control. So, what's happening in the cephalic phase affects the gastric phase, and the gastric phase affects the intestinal phase. And there's a lot of different things happening within the GI system as we process the food and liquid that we consume. This first diagram gives you a summary of the various activities that are taking place within the various regions of the digestive system. So for example, in the oral cavity and in the esophagus, it breaks down the motility, the secretion, the digestion, and then of course if there was any absorption. There's no absorption taking place in the oral cavity and esophagus, so that's why it's listed as none. As you get to the stomach, what kind of motility, what kind of secretion, what kind of digestion, what kind of absorption is taking place, and so forth as we look at the small intestine and as you look at the large intestine. So what I would like to do is go through the cephalic phase, gastric phase, and intestinal phases with you and look at various activities that are taking place as you move throughout the digestive tract. So in the cephalic phase, what we're looking at in the cephalic phase is basically digestion beginning before food ever even enters into your mouth. When you see food, when you smell food, when you think about food, you are essentially triggering those long reflexes that we categorize as the cephalic phase. So we are anticipating the food or liquid entering the digestive system. And so we're getting the body ready for that food or liquid that's about to enter into the body. So these anticipatory stimuli are essentially activating neurons within your oral cavity. They're activating the GI system, the stomach of the GI system in anticipation for food or liquid that's on its way. So you may have experienced that rumbling and growling of your stomach before you actually eat anything. And that's because that cephalic phase is getting everything started. So salivary glands are responding, the stomach is responding, you're increasing your motility and your secretions in response to food that is on its way. So here we have food is coming, or we think about food, and then we see this response, right, as we are affecting the secretion and motility of the gastrointestinal tract. So what are some different things that we actually see when we're in the cephalic phase? And then as we, again, once we start putting food and liquid into the digestive system. So food that enters the mouth is going to be met by saliva because the saliva is already being produced. And it's under the control of the parasympathetic branch of the autonomic nervous system. Because saliva is being put into the oral cavity, it's going to help to soften food, it's going to help lubricate your mouth, and it's going to make it easier for you to swallow. In your saliva, you will have salivary amylase, and that will start chemical digestion of starches that you put into the oral cavity. You also have antimicrobial agents in your saliva, so something called lysozyme is in your saliva that can fight against bacteria. You have antibodies in your saliva, which can disable essentially bacteria and viruses. And so it gives us a form of protection against microorganisms. And then, of course, once you do put food into your mouth, you have that mechanical digestion that is taking place where you use your teeth and you are chewing and you are grinding and you're getting ready to swallow the food material that has entered your oral cavity. Once you have decided that it's time to swallow what you've been chewing, you go through what's called a deglutition reflex. So deglutition is your swallowing reflex, and this is what's going to push the food bolus. So we call this newly ground up material that's in your mouth a bolus. So we're going to push the food bolus into your esophagus. Swallowing begins as a voluntary process. You choose to swallow, and then it switches into an involuntary response of musculature. So basically, you have the ability to initiate the swallowing process. We do that through the glossopharyngeal nerve. That would be cranial nerve number 9. And so we're using information through the cranial nerves. This is cranial nerve number 9, the glossopharyngeal nerve. And we're using motor pathways to initiate the swallowing process that pushes the bolus towards the esophagus. We get information, obviously, from our brainstem. We get information traveling through the central nervous system and then out along nerves. And we're controlling pharyngeal and upper esophageal skeletal musculature. Essentially, the autonomic nervous system takes over, and the lower esophageal musculature and laryngeal muscles can respond in an involuntary way. So when you swallow, the nasopharynx is being blocked by your uvula. So you can see the uvula right here. So the nasopharynx is being blocked. You will also notice that your larynx elevates. So your larynx is going to move up. And what that does is that allows for the epiglottis to cover over the larynx so that the food bolus actually travels down into the esophagus. So here's the esophagus here, and here's your vocal cords where your larynx is located. And you can see that the epiglottis now is sealing off that opening so that the bolus can move down into the esophagus. And again, these are involuntary. Once you initiate the process of swallowing, then you get these involuntary contractions in the deglutition pathway. So the trachea, right, the larynx and the trachea become protected as the epiglottis covers over those openings, and so your bolus can go down the esophagus. Just like the uvula is blocking the nasopharynx to prevent food and liquid going into the nasal cavity. Then you have more autonomic nervous system control, and peristaltic activity is going to move that food bolus down the esophagus. You can see that in that third picture here. We're going to move that food bolus down the esophagus and move it towards the stomach. The lower esophageal sphincter is going to relax. That's also known as your cardiac sphincter, and that's going to allow the food bolus to enter into your stomach. Some individuals have issues with that lower esophageal sphincter, or what we call the gastroesophageal sphincter or cardiac sphincter, and they experience what's known as GERD. GERD is gastroesophageal reflux disease. That's when that sphincter doesn't properly keep the contents of the stomach within the stomach, and so you get regurgitation into the esophagus, and that will irritate the esophagus, and it causes pain and irritation and inflammation. We typically call it heartburn, but really it's esophageal burn, right? You're burning the mucosal lining of the esophagus. So that's the cephalic phase that's leading into deglutition and then sending our bolus down into the stomach. In the stomach, we can transition now into what we would refer to as the gastric phase. The gastric phase is basically looking at what's happening in the stomach and looking at really all the different cells that are involved in processing the food material that's in the stomach. So what are the functions of our stomach? So the bolus has come down the esophagus, and we've got food and liquid that's entered into the stomach. What is the stomach doing for us? Number one, it's for storage. It stores food and liquid, and it's going to help to regulate the passage of that food and liquid into our small intestine. We get some digestion that takes place in the stomach. There is chemical digestion using the enzyme pepsin to break down proteins. There's also some mechanical, of course, digestion, the churning and the mixing of the stomach, and that results in a liquid material that we refer to as chyme. So the bolus has moved down the esophagus, and we're mixing it with stomach acids and digestive enzymes, and so we are liquefying the material, and we call that liquefied material in the stomach chyme. We also have a protective function of the stomach. The acids that are produced by the stomach can destroy microorganisms, so we can kill bacteria, certain bacteria, not all of them, but we can certainly kill a lot of bacteria in our stomach and other microorganisms that are potentially harmful to our body. And so the stomach at the same time has to protect itself from the acids in the stomach deteriorating the organ itself. So there's got to be mucus production and prevention of digestion of the stomach. And so we'll talk about the different cells and what they're doing in the stomach to give you a better idea of what's happening in this gastric phase. So the stomach stores food, right? It's controlling how that food and liquid is released into the small intestine. It's important to do that. If you don't regulate the emptying of the stomach, you're going to overwhelm the small intestine, and that's not going to give the small intestine time to absorb nutrients properly. And what we've seen in some individuals is that when the stomach or the intestine is rerouted in certain ways, or there's been a removal of portions of the stomach or small intestine, this actually can lead to chronic diarrhea situations because you're not naturally controlling the emptying of the stomach into the small intestine the way that it was supposed to be. And that is because we have surgically changed essentially the plumbing of the GI tract. So this dumping syndrome can happen if you're not careful with the actual anatomical structures of the stomach and the small intestine, because they work together to control the emptying into the small intestine. So what are some of the different cells, though, that we see working within this gastric phase? So one of the cells to make note of is the parietal cells. The parietal cells are important in producing hydrochloric acid. So they produce the acids produces the acid in the stomach that we use to kill bacteria and kill pathogens, but also is involved in some of the digestive process as well. The parietal cells also produce intrinsic factor. Intrinsic factor is used in vitamin B12 absorption, and we use vitamin B12 in red blood cell manufacturing. And so without proper B12, so without proper intrinsic factor, without proper B12 absorption, you actually would suffer from a condition called pernicious anemia, which would affect your oxygen carrying capacity because of the lack of red blood cell production. So parietal cells are very important in the gastric phase, making acids and making intrinsic factor. Another important cell is the chief cells. And again, these are all in what we call the gastric glands. So this structure here is the gastric gland. And so we have a variety of cells in here. The opening here is the gastric pit. Okay. So we have pits leading to these glandular structures made up of these cells that produce various secretory products to control this gastric phase. So the chief cells are important because they produce pepsinogen. Pepsinogen is the inactive version of pepsin. Pepsin becomes activated when it's mixed with the hydrochloric acid. So pepsinogen is the inactive form, but then it becomes pepsin, and it's used for protein digestion. Another thing that chief cells produce is gastric lipase, and gastric lipase is used in fat metabolism. That's actually more important usually in your infancy rather than as you become an adult. We produce lipases in our pancreas that are going to be adequate for lipid digestion as we get older. Enterochromaffin-like cells, these are important because they secrete histamine, and histamine is important in the regulation of the parietal cells and hydrochloric acid production. So histamine is involved in the pathway for the production of hydrochloric acid, and I'll go over that with you in just a second. D cells in the gastric glands produce a hormone called somatostatin. The somatostatin hormone helps to inhibit gastric acids and inhibit secretions. You can almost think of it as slowing down the digestive process, which is important because we not only do we need to speed up digestion, but at times we need to slow down digestion. And so it's important that we have antagonistic hormones to aid in that process. G cells, these produce gastrin. Gastrin is a stimulatory hormone. It helps to increase gastric acid secretion and increase motility within the stomach. And so that's going to get the digestive process going. So gastrin from the G cells gets things going. Somatostatin from the D cells slows things down. So you can think of G for go and S for slow. And that'll help you guys remember what's happening with these particular hormones. Another important cell is the mucous neck cells. The mucous neck cells are producing, obviously, mucus, but also bicarbonate. We actually use that to protect the lining of the stomach. So there is obviously gastric acid in the lumen of the stomach, but you still have to be able to protect the mucosal cells so that they're not damaged by the acidity. And so what you see is something like this, where you have a barrier. So in the lumen of the stomach, you've got gastric juice at a pH of 2, so very acidic, but lining the actual cells of the mucosa of the stomach, you have this mucus layer and you've got a bicarbonate content, and that is protecting those cells, and it's keeping the pH at around a neutral pH. So we prevent the acid from actually touching the mucosal lining so that there's not damage to the lining of the stomach. If there was damage to the lining of the stomach, you would see ulcerations. So this particular image shows you the nice, smooth, slick lining that you should see when there's no damage, but then you can see areas that have erosion taking place. That's damaged areas so this is all damaged area of that mucosal lining, and that's because there's probably been potentially some acid damage. Oftentimes it starts from bacterial damage, and then you get into seeing damage caused by the acids of the stomach. So we want to keep that barrier between the acids of the stomach and the actual mucosal lining. How do we control the acid secretion in this gastric phase? So in this gastric phase, you're going to see that you are controlling the acid secretion based on the functionality of gastrin and histamine. So control of gastric acid secretion is based off of these cells interacting together and controlling what the parietal cells are actually doing. So this particular diagram is basically showing you that when you have food, and this also happens obviously from the cephalic phase as well. So it isn't just food entering the stomach triggers acid secretion. We get acid secretion even prior to the food entering the digestive system. So we have vagus nerve input, which is basically parasympathetic, right, activity. Your vagus nerve is the majority of your parasympathetic portion of the autonomic nervous system. So we have input from the vagus nerve, and then of course we also have food that enters into the stomach, and these are triggers for the enteric pathway, the brain of the digestive system. These are triggers for the digestive system to start releasing the chemical signals that are going to help us make stomach acid. So gastrin is stimulatory. So the G cells produce gastrin, and that gastrin is going to affect your enterochromaffin-like cells, and enterochromaffin-like cells are going to produce histamine, and that histamine is going to affect the parietal cells. But you'll also notice that the vagus nerve, right, has a direct pathway to the parietal cells as well. So we're using the parasympathetic nervous system to activate parietal cells, and we're also using gastrin and then, of course, histamine to activate the parietal cells. So gastrin, and essentially it's acetylcholine neurotransmitter from the vagus nerve. They're activating the parietal cells, and they're also activating the ECL cells to produce histamine. The histamine is going to bind to receptors. So if I can actually just draw one here, we'll just put a receptor here. Histamine is going to bind to receptors that are on the parietal cells. These are known as H2 receptors. These are different than what you would see in a pathway for allergies. So these are H2 receptors, but they're responding to histamine in the digestive tract, and that's triggering the production of hydrochloric acid. And so the hydrogen essentially that enters into the lumen is what's making the lumen of the stomach acidic. Acid in the stomach is also triggering the chief cells, right? The chief cells are producing the pepsinogen, that precursor, and then of course it's the acid content that is turning the pepsinogen into pepsin so that we can get protein digestion. If you want to, obviously, if you want to slow this process down, there needs to be an inhibitory pathway. So as acid production increases, so that as there's an increase in hydrogen ions, and so the pH is getting lower and lower, as the pH drops, you're going to see the activation of D cells, and D cells produce somatostatin, and somatostatin is inhibitory. So somatostatin negatively inhibits, right? It inhibits the G cells from making gastrin. It's blocking the histamine from interacting with the parietal cells. And so you will begin to slow down the acid production. So you have both stimulation for acid production, but then if you start to get too acidic, you have a pathway that's going to decrease the acid production. Balancing out acid production obviously is important so that we prevent having ulcerations in the tissue. There are different sort of ulcer pathways. Some of our peptic ulcers or duodenal ulcers are caused by bacteria presence. It's called Helicobacter pylori, H. pylori for short. H. pylori is a cause for a lot of ulcers that occur in the digestive system. This bacteria causes inflammation. It affects the gastric mucosa, and then acids can get into those spaces and deteriorate the lining. We also have conditions like Zollinger-Ellison syndrome. Zollinger-Ellison syndrome actually results in making too much gastrin, and we know that gastrin is stimulatory to acid production, and so that can lead to hyperacidity, and that hyperacidity also leads to ulcerations in the GI tract. Some other culprits for ulcers would be overusing NSAID medications. NSAIDs are non-steroidal anti-inflammatory drugs. These would be things like aspirin. And so overusing NSAID drugs can actually cause some damage to the gastrointestinal lining. And then, of course, as you get damage to the lining, it opens up space for acids to seep into the tissue spaces and cause deterioration. So how do we treat ulcerations? Oftentimes we treat ulcerations, sorry about that. We treat ulcerations by controlling the bacterial. So go back here and write H. pylori again. We control the bacterial population. So we can use antibiotics to control the H. pylori infections. We can use medications that reduce the amount of acid that we produce in the stomach. So you may be familiar with things like Tagamet or Zantac, Prilosec, Prevacid. These are medications that can inhibit the production of hydrochloric acid in the stomach and therefore control acid production. The last phase that we want to look at is the intestinal phase. The intestinal phase is basically slowing down the stomach so that the intestines have time to do their job. So in the intestinal phase, it's basically a series of reflexes to feed back on the stomach and control the emptying of the stomach so that we control how much chyme enters into the small intestine so that we have proper time to digest and absorb nutrients in the small intestine before we receive more material from the stomach. So it's basically feedback pathways. So for example, when chyme enters into the small intestine, we're going to have the nervous system feeding back. So the nervous system, the enteric nervous system. Right? So as chyme enters into the small intestine, the enteric nervous system is going to feed back, right? It's going to go back and inhibit gastric motility. It's going to slow down the stomach and give the intestine time to process what has entered into it. So think of it like this. If this is the stomach, right, and you've got a whole bunch of chyme in there, and then this represents the beginning of the small intestine, so this is the duodenum, we need to allow, let's say there's 100 milliliters, right, that comes into the duodenum, but in the stomach there are, liters, right? So let's just say there's 1,000, milliliters currently in the stomach. You don't want to overwhelm, right? We don't want to dump material into the small intestine and overwhelm it. You can't dump 1,000 milliliters. Maybe you can only take 100 milliliters at a time. So we want feedback pathways that are going to inhibit, close off the pyloric sphincter, right? Slow down the motility, inhibit the stomach from emptying so that you have time to process, right? We want to process what has entered into the small intestine so that you get proper digestion and proper absorption. So the enteric nervous system is involved in that. And then, of course, hormones are involved in that. So cholecystokinin inhibits gastric emptying, gastric inhibitory peptide, glucagon-like peptide. These are all hormones that can slow down, even secretin, right, can slow down gastric emptying and give the intestine time to process what enters into it. So secretin, cholecystokinin, gastric inhibitory peptide, glucagon-like peptide, these are released to slow down and inhibit the stomach so that the intestines have time to do their job. And that, in essence, is what the intestinal phase is. So secretin is releasing it's stimulating the release of bicarbonate. That's neutralizing acids that are coming out of the stomach. Cholecystokinin is inhibiting gastric emptying, but it's also triggering pancreatic enzyme secretion. It's triggering bile secretion so that we can digest and process properly in the intestines so that we can absorb nutrients. Glucagon-like peptide and gastric inhibitory peptide are slowing down the stomach, but they're also triggering insulin secretion so that we can properly get sugars into our body cells and utilize those for making ATP. And then we even use osmoreceptors to monitor the sort of hyperosmotic conditions that are happening in the intestine, and that will trigger pathways that slow down, right, gastric motility, slow down acid production, and so forth. And so that, in essence, is what the intestinal phase is all about. So we have the gastric phase, we have the cephalic phase, we have the gastric phase, and then we have the intestinal phase, so that we can properly process what's happening in the digestive system. So this last slide is simply a summary of things that we were just talking about. So what's happening in the cephalic phase, what's happening in the gastric phase, and then what's happening in the intestinal phase. So just a quick summary: we think about food, we smell food, we see food, and that is starting, right, the gastric motility and the acid production and so forth. In the gastric phase, we are processing material that enters the stomach. We are making more acids. We are using hormones. We are protecting the stomach. Right? We are doing the things that we need to so that we can make the chyme within the stomach. And then we are releasing that chyme into the intestine, but then the intestine responds through a neural inhibition and through an endocrine inhibition, so that we can, through a hormonal inhibition, so that we can limit gastric emptying and give the intestine time to process what has entered it, so that you can digest properly and absorb properly without pushing too much chyme into the intestines. Because if you push too much chyme into the intestines, that's going to lead to a diarrhea situation. So that is the cephalic, gastric, and intestinal phases of the gastrointestinal tract. I hope that helps. Take care.